Wonderful. Hello, everyone, and welcome to today's CellProfiler workshop. I'm Nasim Jamali, a postdoctoral researcher in Dr. Ann Carpenter's lab in imaging platform at the Broad Institute of MIT and Harvard. And I'm here today with my colleague Pearl Ryder and Beth Cimini. A couple of notes before we start. I'm glad you all have CellProfiler-Analyst already downloaded, and make sure you have the exercises and data sets as well, which you can find the link at the bottom of the slides, along with a PDF file of these slides. Also, mute yourself, please, your microphones while not speaking, and feel free to raise your hand, ask questions anytime during the presentation, unmute yourself and speak up. This is a small group, and we would like to make it as interactive as possible. For exercises, when we get to that point, we'll send you to a small breakout rooms to work together. And for that, if possible, please turn on your cameras and mics and try to work together. And when we get to the demo part of the CellProfiler, I'll be switching back and forth between the slide deck and CellProfiler demo. As we know, images contain a lot of information. You often have millions of pixels per image, and pixels carry digital values. When you have multiple channels and sequence and time points, that gives you a very large set of information on the level of richness of information that you could get from genome, with the additional benefit that you can get information about a spatial organization and see which structures are present in which individual objects. Like how close are these red dots, for example, to the nucleus. These sort of data is hard to get from most of the single cell technique, and a lot of these information are not typically being used. So we are going to show you a set of tools that you can detect the objects of interest so you don't lose any valuable information from your images. This is an image from the dataset we are going to work on today. Here you can see GFP tagged protein, and we want to see whether it's localized in cytoplasm or in the nucleus. Just by looking at these images, we can have a sense of its cytoplasmic or nuclear localization. But if we were looking only at the protein level or mRNA level, we wouldn't be able to detect any localization changes. But no one wants to sit down and analyze images individually one by one. So automatic image analysis, that what we are trying to do here, is objective, quantitative, and is faster and less tedious. And you can do a lot better with your time than counting spots by hand or looking at cells and trying to pick up a phenotype by yourself. Especially because human brain is not very good at quantification. If you look at this image quickly, it looks like a full color image. But when you zoom in on it, you'll see that it's actually a grayscale image, and our brain feels the rest of it. Our brains are designed for taking information quickly and trying to create a scene, and they're not actually great at picking up details or quantification. Another interesting example, if you look at these 4 similar color bars, and now see what happens when we pass a different color lines through these purple columns. They appear to be totally different colors. The segment that compose the waves appear to be hot pink, gray, purple, and beige. But they're actually all the same color. So you are better off letting a software that is designed to do image analysis do your quantification for you, because your brain is constantly trying to trick you. There are a lot of image analysis tools out there, and we are not claiming that CellProfiler is the best software for every job, because we know that there are a lot of good tools out there with advantages in some areas. Most of you have probably used or heard of ImageJ or Fiji. It's very fast, has a lot of plugins almost for any experiment you want to do. The disadvantage, however, is if you want to apply analysis that you designed in Fiji to a lot of images, hundreds or thousands of images, if you're not familiar with writing a script or macros, that's going to be very tricky. So although it has a lot of analysis power, it's not always the best tool to make your analysis reproducible. Another great tool that we like is Elastic. It's very good for identifying images that doesn't have a nice bright fluorescent signal. By picking part of the image as foreground and the other part as background, we basically create a digital stain, and we create different classes. And from those classes, then the computer learns basically which pixel is for which group, foreground or background, and then it can create these sort of digital stains. This is great, and it's something that CellProfiler doesn't do. But the disadvantage is that it does help you to find the object, but it doesn't tell you a lot about the object. So sometimes what we do, we combine the programs together. And what I mean is that we create these digital stains in Elastic, and then we take them to CellProfiler and make a ton of measurements from the objects that are detected. So if you really want to find objects in high throughput, reproducibly, and learn a lot about them, for that, our lab has written CellProfiler and CellProfiler-Analyst for about 15 years now. These are the two halves that can be used independently, but they work the best together. CellProfiler is designed for image analysis and quantification. It's designed to take an image and turn it into a series of measurements from the objects that are present inside it. Once all of that data is extracted, CellProfiler-Analyst is designed to help you to look at comparisons between different treatment groups or different wells in your experiment. And it allows you to see what is going on in your data in an interactive way. And these are both free open source softwares that are available for Windows, Mac, and Linux. This is our software engineering team who done a great job in making the software over the years and making it so that it's the most flexible and user-friendly image analysis software for biological image analysis designed specifically for biologists. Our lab motto is to measure everything and ask questions later. And the CellProfiler is typically the tool we use to make the measurements. The idea is that the harder thing in image analysis is finding the objects that we care about, or the things basically that we care about. And after we've done that, we may as well measure every property of the objects of interest that we can think of, whether we are planning to use it now or no. Because then we may later come up with an idea to measure something else in our analysis that we haven't thought about it before. So then we would have all the measurements required. We just can check the data and do our analysis. So CellProfiler is designed to give you an interface to be able to find these objects no matter where they are, and then take a ton of measurements of them. And then you can go through and decide for yourself later which one to use. CellProfiler is originally designed for high content screening, but certainly it's not the only use for it. It can even be used for 1 or 2 images. The idea is that when you get to the experiment that you have multi-well plates, You can then use CellProfiler to find the objects of interest, for example, cells and nuclei, and create measurements from them across all different conditions and wells in your experiment. And then you can run them through CellProfiler-Analyst, and which is a great tool, and we're going to learn about it later in today's workshop. For example, CellProfiler-Analyst has this very nice plate view, and you can basically look at the whole plate at once. If a feature, and we can see like if a feature of interest is up or down in certain wells, or we can see, look at the scatter plots and, for example, see which wells are outliers. Again, we don't need to have a whole multi-well plate to do this. We are going through this workflow today with only 26 wells. And even if you only had a few images, you still would be able to do this, to use CellProfiler. Let's now think about how do we actually tap into in image analysis workflow. The workflow for analysis is really two steps. One is identifying the objects that we care about, and then make measurements from them. So as we said, the hardest part is figuring out how to find the objects we care about. So for example, here in these two top images, in the right, we have the yeast pound, and which has these round-shaped colonies. These are round, like the DAPI stain nuclei on the left image, but this image is 3-color versus 1, and the colonies are various sizes versus in nuclei, all of them are pretty much similar sizes. So how do we come up with a pipeline that can find the objects that we care about for anybody's individual experiment? Because there are many kinds of biological experiment as the biologist working and creating microscopy images. We don't actually want you to start from scratch on this because this can be tricky. If you go to our website, we have a lot of example pipelines that goes through and teach you how to analyze different cells. For example, human, fruit fly, etc. You can scroll through the images that are there and find the ones that are mostly resembles with the images you are trying to analyze, and pick the example pipeline and start from there. Okay, now let's start and think about what we are trying to do in today's first exercise. This exercise is from real data, from real experiment. And the main question here is how much drug, in this case wortmannin, is needed to cause translocation of this green GFP signal, which is a translocation factor, to cause it to translocate from cytoplasm to nucleus. You saw an image from this data set earlier. The stainings are GFP FOXO1A, and they wanted to find the minimum dose of wortmannin that could cause this translocation to happen. They knew that the translocation happens at 150 micromolar dose of wortmannin, but this dose was very toxic. So they did some titration screen to find the best IC50 that you would need to get translocation to the nucleus. And by the end of this exercise, you'll see how much of this drug is needed for this translocation to happen. Okay, now I'm gonna start CellProfiler and switch back and forth between the slides and live demo. Before I do that, I wanna ask if there are any questions. I don't see. Any questions?
Yeah, sorry, I have a question.
Yeah.
Do we get the slides emailed afterwards, or?
They're already available on the link.
Ah, okay. Oh yeah, wait, I saw that. Sorry.
Okay.
And I see your recording. Do we get the recording afterwards?
I believe I2K workshop folks gonna share it online on
Great.
your website probably.
Okay, thank you.
Sure. Any other question? Okay. So let me share my screen again. If you double click and bring up CellProfiler, you'll see at first this very nice welcome screen. It has different examples, how to get started, some of the examples from our website, tutorials, the manual. It's a great help. It has basically help for every single module and everything that that is changeable basically in CellProfiler. And Q&A forum is the image.sc forum that you can always ask questions and get help from different softwares, including CellProfiler, which I'll talk about later. But we don't need this screen, so I'm just going to close it, but it's always there when you open CellProfiler if you need assistance. Okay, let's make it this way. So CellProfiler has these, maybe this would be easier, has these left and right panel when you open it and look at it. Basically, that's how the interface look like. We call the left panel pipeline panel, which displays modules in the pipeline. And modules are executed in this order that we put it back to back. We're gonna build this today, so don't worry if you don't have the rest of it and only these 4 top part. We'll get there. And on the right, we then have all the settings for all of these individual modules. For example, for overlay outline modules, we can see how to basically show the overlay. What would be the name of it? What object do we want to show the overlay for? What color we want to select, etc. For every individual modules that you see, we have this setting panel for it. These settings, again, are different, depends on which module we are using. It's always this left-right window, a step of the workflow on left and configurations on the right. Now let's go back to the left panel. The first top modules are called input modules, and they're responsible for bookkeeping. So as every experiment is different, CellProfiler doesn't know whether you use 2 channels or 5 channels, or if you have Z-stacks and time points in your imaging. So here we need to adjust these parameters and teach CellProfiler what you were doing during imaging. These steps are very important, so everything after it can work smoothly. And every pipeline has these 4 top modules with some very rare exceptions. After these 4 modules are the analysis modules. These are what you need to tune in and adjust to be able to run the analysis. This is where basically your biology expertise comes to play. And we can add, remove, or reorder modules with this button in here. And I'll show you in the live demo in a second how we do that as well. Okay, let's switch to the live demo now. And what I want you to do is from the folder that you downloaded, the translocation folder, it shows these 4 files in that. We have the translocation start and final pipelines, along with the translocation PDF and the data. So now I want you to drag and drop the start pipeline into the pipeline panel, into the left panel here. When you do that, it's gonna ask you, do you want the load pipeline? And you say yes. So it only has now the 4 top pipelines and identify primary object, but we're gonna build it together and move forward. You can open the translocation PDF file, and we're gonna follow along with this PDF file. And then drag and drop the translocation data. These are all the images in our data set into where it says drag files and folders here. So I'm just gonna drag and drop it here. Now let's try to figure the settings for these input modules out together. If everybody so far is here.
I know that we did have some folks had some issues unzipping the file, so they may not be able to follow.
Okay, then let me briefly explain here, and then you can work together in breakout rooms. Because these are important information I want you all to know. When you double click in one of the files, you can actually see the images. A couple of, there is a very nice toolbar on the top. You can zoom, you can click this magnifier icon, and you can zoom on your image. A step, backward step takes you one step back, and forward takes you a step forward, and home takes you to the actual unzoomed image. We can also use this measure length tool and measure the length of these nuclei, for example. And you can see where that shows in the bottom here. So when I move my mouse here, you can see that it shows the location of my pointer and also the intensity of the pixel in that spot. And when I use measure length, you can see that it shows length as well.
Sorry, is that in pixels, the length?
Sorry, yes, it is. It is.
In pixels, okay, thanks.
Another nice thing is you can right click here and adjust contrast, and it has different modes. On normalized, you can just adjust the brightness. In log normalized, you can adjust it based on log. Normalization factor, if I click OK, it's gonna change these. On the modules after these four input modules, the panel gonna change to 4 different sections, and you'll see that for these adjusting contrast, there's gonna be a button asking you if you wanna change it for all the panels, and you can say yes or no. One more thing here is when I right click and I can see the histogram for this image. And what it shows is basically every single pixel, intensity of every single pixel in this image. And we're gonna show you this histogram later when we talk about thresholding and how to detect the objects that we care about. Okay. And in the folder as well, we have a CSV file, which I'll get to a second. Why do we have that? Sometimes for your experiments, you have different doses of drug, different treatments, and sort of like a metadata of what you did in that plate. And that's always helpful to bring it to metadata along with how we extract information from the file name. So we don't need to, you won't need to go back and forth between, okay, which well was what dose, et cetera, et cetera. So now the next step, we want to extract information from our files. As you see here in the file names, I can see that there is information about the well, about the site. This is only one site per well, but you may have like 10, 15, depends on your experiment, different number of sites that you imaged per well, and also different channels. Here we only have 2, but you may have 5, 6, or more. So we want to extract this information. And how we do that is in metadata by extracting from file folder names. Now I want to extract from file name. And this is basically a text expression that that's a regular expression editor. If you click and choose the name of one of the files and paste it here and click yes, usually CellProfiler does a very good job of detecting and extracting information, but it depends on your instrument and how the naming system is. It may not work. So that needs a little bit of getting used to. You need to see a couple of examples to figure out, okay, how to exactly extract the information. Here, what we have is plate name, well, site, and channel number. What we care about here, because all of them are one site and it's all in one plate, basically for our exercise, we only care about well and channel number. And we do have that as well. So submit. The next extraction method, I want to bring in the CSV file. Let's take a look and see what information are present in the CSV file. So we have the well information, the dose of the drug worth manning, and whether that well is a positive control or negative control. And there are some wells that are showing 0.5 for positive, negative, and those are our different doses of the drug that they want to find what's the best dose for the translocation to happen, the minimum dose that can happen. And we want to bring this information. So what you need to do now, if you have your CellProfiler open and running, to make sure that you are going to the right folder. So now, for me, it wasn't in the download file. For me, it's in a different place. So you want to find where you basically have your files, locate the CSV file on your computer and open it. And then you want to make sure to match the well information. So the well information in my CSV is the same as the well information in the images metadata. So I'm going to make sure they are matched. And if it's already not set, make sure to have the dose data type on float because it has decimal points. That helps. And before you update, if you take a look, we only have well information and channel numbers, which were extracted already from the file name. But when I click update now, we can see the different doses and also whether it's a positive control or negative control. For those of you that are following along, are you here with me?
Yep.
Perfect.
Yeah.
Awesome. The next step.
Somehow it didn't work for me. I mean, I have the positive negative controls there, but all of them have the value 0. So something must have gone wrong.
All of them shows value 0. Can you make sure that you have the CSV file? I mean, open the CSV file, make sure the file is okay, and also the location of it where you set it. And the information.
That looks all good.
Okay. I'd say if you don't mind, let me continue. And when we go to breakout rooms, I can stop by your room and double check.
Yeah, sure.
Thanks. All right. The next step now is teaching CellProfiler which channel is what. So we have W1 and W2 here that basically distinguishes these two channels from each other. So I'm gonna tell CellProfiler the file that does contain W2 is my DNA channel, and I already know that. And the file that does contain W1 is my GFP channel. and just update. Now we created an image set. So for CellProfiler, for every channel that we have in our data set, for how many channels that we have, we want to make all of those as one. We call all of those together one image set. If I have 2 channels, those 2 channels are my one image set. If I have 5, those 5 channels are my one image set. And what that means is I'm gonna run all of those, or I wanna have access to all of those at once to run it through CellProfiler. And if I go back to metadata, I see 52 rows, which are all of the files that are here. But if I go to names and types, and when I hit update, I see half of those numbers because I have 2 channels, and both of these channels together are making one image set. Don't worry about the groups here, just set it as no. But in general, what the grouping does, if you have, for example, 10 plates, you could group your analysis per plate, or if you have more than one site per well, you could also have group them per well for analysis. Let's make sure I covered all of these, and then before we move forward to actually putting some analysis module together.
Can I ask about the
Yes.
grouping?
Yes.
Can you then also, for example, have different thresholds for different groups, or does it still run the same analysis on the groups?
The same analysis on the groups, but the thresholding basically based on the algorithm that you choose, which we're gonna get there in a couple of minutes.
Yeah.
That how we set the thresholding, and then after that, it's gonna be basically the range that we have for thresholding is gonna be the algorithm is the same. The way that we do thresholding for all the analysis are the same. The only thing is that the exact thresholding, basically the threshold is gonna be slightly different, but in the range of what we defined for every single image, because every image has different pixel values. And based on the algorithm that we picked, it's gonna change slightly.
Okay.
Wonderful. I wanted to show you this. So here, if I want to add a module, I click the plus button, and we have the module categories for file processing, image processing, objects, etc. And this is very nice as well. I can just write as well, identify, for example, primary objects. It's search based, so I can just search for the name of the module. If you are not familiar with the name, so then the categories comes helpful. You can go to object processing. After primary comes secondary, so I'm gonna add it to the pipeline. Now we don't have it set up, so you can see that CellProfiler is not happy here, and that's another very nice method that is talking to you. It's telling you I'm not happy, something is not set properly. If I want to move the modules up and down, if I want to delete a module with this minus button, and when we have a series of modules like here, we can turn the module on and off. So when we are testing our module still, we may have additional modules, but when we want to run the analysis, we may decide not to run some of them, or when I'm testing, sometimes if a measurement module takes long, I can turn it off and only test for identify primary objects, only make sure that my detection and segmentation works well before running. So I could easily just deactivate the module. And what this I button does, when I go to test mode, we see 4 different buttons here. Again, we get to work on these in a second, but I just want to show you the purpose of the eye. If I click a step, a window gonna pop up, and it's actually showing this window what the eye does. If I close it and hit a step, it won't show the window anymore. For the test mode, we want to make sure that we have the eye open, and a step basically moves in the test mode one step, one module at a time. If I click the pause button, so if I have like 10 different modules here, if I put the pause button on before the measure colocalization, for example, and hit run, it's gonna run all of the modules up to the point that I put the pause button. Then I can make some adjustment and hit a step. And it would show me the window. The next image set is basically just choosing the next image set in your data. And when I'm in test mode, I could actually go ahead and choose my own image set from the data I have. And I could even choose a random image set and randomly picks one of the image sets to run through the pipeline. When we are done setting up and we are happy with our pipeline, then we want to make sure to set output setting. That basically tells us where do you want to save your analysis file. So you can, after we are done putting the pipeline together, you want to make sure that you choose the correct location or wherever basically you want to save your files. And then after all of it is done, we're gonna close the eye, and the best, when you have a lot of modules, we don't wanna go one by one and close. You can go to window and close all windows, hide all windows and run, and it's gonna hide all of the windows. What close all open windows is if I'm testing and I have like several windows, like you saw for identify primary object open, it's gonna close them and clean your window basically all at once. And hide all windows, and when everything is set, then we click analyze images. We are not there yet, but just so you know. Let's make sure I have all of these. I did talk about the image set, and after we basically set the image set, we go through and add the modules for image processing, object processing, measurement module. And then at the end, we want to have file processing to basically save all the, if you need to save all the images, and you want to make sure to save all the measurements, either to an Excel file or to a database. This is important because CellProfiler doesn't really just go ahead and save everything. You need to add the module for file processing to basically save everything you need. Let's see. Okay, we did cover all of these. Let's go and send you guys to break. Oh, I want to actually talk about how do we go ahead and basically do object identification before I can let you play with the setting in identify primary object. So a couple of more slides, and these are very important. And identify primary objects is one of the most important modules because it's basically setting the base for all the other modules after that. So once the images are loaded, how do we actually find the object of interest? How many nuclei do you see in this image?
2.
2, yes. So 1 and 2. But how do we really know that these are 2? I mean, how can we distinguish these? Object analysis is usually done in 2 steps. The first step is distinguishing what is the foreground from background. Basically, what are the stuff that we care about versus what we don't care about. And then we need to figure out how to split these objects or merge these 2 objects, depends on what we want to do. And this is done in Identify Primary Objects module. One of the important features to define here is thresholding. For almost any thresholding algorithm out there, we look at the intensity histogram that I showed you earlier for an image. And that's basically what CellProfiler does. It looks at the image histogram, intensity histogram for every individual image as they go through the pipeline. So every image has pixels, and each pixel contains digital values or numerical values. And most image histograms have something like this, a background peak with low intensity, and the stained objects that are showing with higher intensity or second peak. The goal of a thresholding algorithm is basically where to draw the dividing line between where the stuff is or where the background is and where the stuff that we care about. If I draw a line here, we capture the nucleus, but we also capture a lot of background, which in this case is cytoplasm. So now my biological question is I want to detect nuclear translocation. So I don't want to have an area that contains both cytoplasm and nucleus. So this is not a good threshold to draw. Also, if I draw too high of a threshold, I don't have any background or cytoplasm, but I also didn't capture all of the nucleus either. So we always look for the best threshold that can capture only the object in question and nothing more. Since there are different methods to draw this thresholding line, there are several methods that are already part of the CellProfiler, and you get a chance to play with them all. But I just want to briefly introduce some of them. MCE, minimum cross entropy, is our default, and it's great when you have a good divide between the foreground signal and background. ODSU is very nice. It gives you 2 or 3 class option. So if you have 3 classes of intensity instead of 2, for example, when you have background, autofluorescent, and cells, you might have 3 bumps in the histogram, but you only care about the top 2. Or when you have background, dim cells, and bright cells, you also have 3 bumps in the histogram, but you only care about the top, the top 2. Or in this case, I mean, you can basically choose where to send the middle peak to be part of the foreground or the objects you want to detect, or the background, that what you don't want to care about. And then another option is robust background, which is great for a speckly object. And if anyone here works on fish experiment, where most of the image is background and less than 5% is actually signal, robust background does a very nice job. And these are all located here in Identify Primary Object module. And you can read about every individual module and every setting from the help button. So if you are on the help button, if you have the module highlighted, when you click help, a window pops up and it shows you every single setting and a lot of information about the module. If you click on the setting part of the module, for example, I want to know what robust background does, you can read about it here. We do believe that CellProfiler has the most help buttons and basically most information about every individual modules and settings. Okay, next. To tweak the threshold, you can apply the correction factor. If your threshold is always a little bit too strict or too lenient, you can apply the correction factor. For example, we can multiply thresholding by 1.5 or bring it lower by multiplying it to a smaller number. We typically want to stay close to 1 because we do want to trust the algorithm that was picked for the analysis. But if the cell profiler is always a bit too strict or a bit too lenient, then you can adjust this threshold. You can also set upper and lower bound threshold, which is really nice when you have, for example, some wells that are empty or some wells that have some artifacts in them. It's not really the case in this experiment we want to work on today, but it always basically allows us to gate off part of the histogram and tell the algorithm to only choose the thresholding line between these two numbers. And don't let it to draw the threshold beyond a specific part or basically, for example, center section that we chose. And these settings are here, the correction factor and lower and upper bound threshold. The next question becomes, okay, now that we figured what is part of our foreground and what is background, then we need to separate any objects that might be touching. In this image that you see, how many in this central clump, how many objects can you count? 1, 2, 3, 4, 5, 6, 7, 8. How do we know? One way to tell is that there are 8 objects here is the brightness. Fluorescent stained cells tend to be brighter in the center and they get dimmer at the edges. So you can see here, the signal is brighter here and dimmer here on the edge. Also, after so many years of looking at a cell, I know that how a cell looks like this, for example. And these two, to me, looks like two different cells that are basically next to each other. So what distinguishes that is shape. So what we just talked about is basically declumping method. And how do we do this is exactly what we just talked about. Is basically first based on the brightness of the intensity at the center and also the shape of the object. And these settings, I'll show you in a second where they are located. But basically here, the brightness are the peak of these mountains. So these are the highest intensity or the brightness for these 8, or this central clump. And then also we have another method to say that, okay, just separate these objects based on shape. So again, that goes through the same thing, which is basically having these high intensity peaks or based on the shape. So when there are 2 objects touching each other, if the intensity is not the case, then you can separate the clumped object based on the shape. Since for example, for cells, a lot of biological objects are roughly circular, such as nuclei and cell. You can tell CellProfiler to look for something that is roughly circular shaped. And if there are indentations like this hourglass shape, to count these as 2 objects. Then the next question becomes where to draw the dividing line between the clumped object. Again, the settings are based on shape and intensity. So we know that, again, biological objects are roughly round, and we can use that and say, just pick a middle line between these 2 roughly circular shapes and draw a dividing line between those. Or we could follow the intensity boundaries. and see where the intensity falls off, and then draw the dividing line between these two objects where the intensity falls off. You can use these both for detection, for separating, and also basically separating these two objects and putting the dividing lines in the combination between shape and intensity. So for example, use intensity to find the center and then draw the dividing line based on the shape or any other combination. You should always test both and see which one works better for your images. And these settings are here. Method to distinguish clumped object and draw a dividing line. Sometimes it happens that when we go through the first round of testing the identify primary object, we see that the segmentation of the objects are splitted incorrectly, or they are merged together incorrectly. So there are two more separation settings that you could check. The first is how much smoothing we apply to the image before break them into the pieces. You see, for example, this nuclei is a bit crunchy. It has some sort of a bright spot here on the top half. And this is something that is pretty common in a lot of objects, especially in the mouse nuclei. They tend to have these bright heterochromatin blobs. So in this case, we want to smooth this image before basically detecting the object. So we don't end up having these two separated nuclei when we know they are only one. So if I set the smoothing filter size to 4, it's not smooth enough, so it would separate those. But if I increase the size and make it to 8, it would detect this whole nuclei as 1. However, it could be that in another image, or even in the same image, another part of it, the setting that works for another image, it doesn't work for another part of the image. So this smoothing of 8 is too much here, and we see like a part of the nuclei here, and it does merge with the one next to it. This does happen, and if you have this sort of situation, our rule of thumb is that you get similar number of merged one and a split one. So don't spend hours trying to get everything perfect. It's never gonna happen. Another setting that we could also check is the suppression of local maxima. Here, again, we can use our biological knowledge about the cell and tell it how close any cell centers can be from to each other. So you see these are the two bright peaks in this nuclei. And from experience, we know that nuclei has certain size. So any two dots that are, for example, here 4 pixels away from each other are probably 2 dots inside of the same nucleus, rather than 2 separate nuclei. I can tell it to ignore areas that are closer than 4 pixels apart. And if I set the minimum distance between the local maxima to 6 here, now it knows that these are from the same nuclei, and it'll segment this nucleus correctly. Again, the major thing with all of these settings is, and the combination that you end up choosing, is that there is no perfect combination that we can find every object perfectly. If you have this sort of situation, again, try to balance it. Have the roughly similar number of breaks and merged if you can't get it all perfect. And these settings are here: the size of a smoothing filter and the allowed distance for local maxima. The very last setting, before you all get to play with the settings yourself, is the object size. Here again, based on your knowledge, your experiment, you know the object that you're looking for is roughly what's the size of it. Here you can define a minimum and maximum diameter of an object and say If an object is bigger or smaller than a certain size, you don't think it's a real object. And you don't want CellProfiler to include it in the analysis. For the setting for object touching the border of the image, if it depends on what type of measurement you want to make. If you basically want to measure the cell size, for example, then you don't want to include the objects that are touching the border. So you don't want to have half or part of a cell. But if your object of interest are rare and only like dots inside of the nuclei, you may want to include everything. Depends on, again, what you're looking for and the question you are trying to answer. A couple of more things here is I go to start mode and a step. The objects that are showing, the segmentation gonna show in different colors here in this window. The objects that are picked, basically, what is passed through the identify primary object module is going to be shown here with green outline, and everything with this pink outline is they are the invalid size object. They are not accepted because of size, and everything that you see on the borders for like this orangish yellowish color is basically the ones that are not picked because they were discarded, they were touching the border of an image. One more thing is when you zoom in, you have this pan button. You can move around in your image and check different parts of your image. Alrighty, with that, I think now we are going to send you to the breakout rooms. If you have any questions, please feel free to ask now. Otherwise, follow the PDF file, work in groups, and try to set the best settings for the identify primary object module, and follow along on the PDF. Any questions so far?
If we could maybe get those who are still having technical issues to let us know right now, and we'll create a separate breakout room with Beth to try to figure out those technical issues. So I know some folks were having issues unzipping the folder. Is that still going on? Breakout rooms, Nasim and I will be moving around, visiting your rooms, so we'll be available to help as you go.
Yeah. And let's have about 15 minutes for this. And we pop in and out if you have questions. Okay, off you go. And Beth, how was it?
I still didn't manage to figure out how to make two nuclei that were very close to each other to be split.
Yeah, we had the same problem.
Did you, a couple of things is don't rely only on one image. Try to test several images when you're testing your pipeline. And you can either click next image set to test, or you could basically from the test button that I showed you, let me share again. You can test on several images, and also, so here you can test next image, try several, or go to test and choose random image set, or actually bring up your image set and just randomly click on a couple to see how your thresholding work. Again, this is the first pipeline, so I haven't played with it, but that's how it would do. And then if you follow the PDF file, all the instructions are there. But again, you would have another opportunity to work on that and the rest of the pipeline again. Okay, I'm gonna exit test mode now. And we want you to build the pipeline from the start to get to the final. But I'm just gonna load the final now. So I have all the completed pipeline. Again, I wanna make sure that I have the metadata, the folder, that I have my CSV file there. Make sure that that's selected. And then click my CSV file, make sure the rest of it is fine. Update, I have the dose and the names and types. I have 26 image sets. So that's the whole pipeline. If for any reason you run out of time after this section, you can just load, and you couldn't basically build all of these modules back to back. You can just load the final pipeline, make sure to do the adjustment, make sure to have the output setting set to the location you want to save your analysis data. You need to know where that is, because we need to extract a file from it for the analyst later, and then click Analyze Images. But that's for later. Let's now go back to the rest of our presentation. Let's see. Okay, we did cover the primary object. The next step, when you get to identify secondary objects, there are again a couple of different methods that we can pick. Distance n, for example, or let me show you, there are a couple of different ways. We have distance n, we have propagation, watershed, and distance b. And what this does, again, it really depends on what you choose, depends on your staining and your biological question. Distance n, draw equally sized circle around each nucleus. This is useful when you don't have another cell stain that you can use this reliably to determine the cell boundaries. There are other methods. Watershed, propagate, distance b. Distance b uses combination of distance from the seed object and the intensity that is present there. So we want to use a method that here for our experiment, for our question today, treat both positive and negative control the same. So that's why we are suggesting distance n here, because there are like half of the images here that they don't really show any GFP in cytoplasm. So it would be hard to detect the cells. But again, what you choose depends on your goal and your biological question. And then the tertiary object is basically subtracting the nuclei from the cells, which leaves you with cytoplasm here. And for measurements, what you, again, pick and choose, it depends on what you want to answer. Here we want object intensity, and we want to make sure that we have it both for, we want the GFP in both cytoplasm and nuclei, because we want to see the translocation and how it goes. We could also measure colocalization in different objects and from different channels. If when you're adding colocalization, make sure to have the method for Costas thresholding to the faster mode if you're running all matrices. But again, these are, you can read more about it on the manual from the help button, what each of them do. But when you get to play with a lot of different measurement modules later on, you figure which one would be helpful for your analysis. And then we are calculating a ratio basically of what's the intensity of the GFP in nuclei versus cytoplasm. You can see all of these here. And the important part, again, is the data export modules. So CellProfiler by default doesn't save anything other than the pipeline itself. And even for the pipeline, I'll show you that there are two ways to save it. The goal for the export module, data export module, is to save measurements later for the analysis and whatever you need to do with the measurements you made. There are several options: export to a spreadsheet and export to database, which from the name it's obvious. And we can save images intermediately, like whatever you picked. For example, for your primary object or secondary object, you can save all of those in save images. In here, we only care to save the overlay from the nuclei. And I had a question earlier that how do we tell each module which image to load? Basically, in the top of each module, there is this input image that you can set which image to choose, and it tells you where did it pick that image from. So in names and type, we named all the W2 channels raw DNA, and that's where it basically loads and reads the image. And here, because we later want to work with CellProfiler-Analyst, today we are exporting to database because that's the file that we need. But usually you could just send it to a spreadsheet. And how to save your pipeline? You can save the project, or you can export the pipeline and make it more portable. What that means is when I want to save the pipeline, I can go to File, Save Project, or Save Project As, and save the project. And what it does, basically, it has the file, the location of my data, and every setting that I set already. So it has the images location and metadata that we set. And it's a very good quick starter. So it means that if I'm done with the day-to-day and I'm still not happy with the pipeline I put together, I can save it as a project, save it on my computer, and tomorrow morning, if I'm working on the same system, just drag and drop the CP-Proj file to the pipeline panel, and then it already knows where the data is, where the metadata is, and so on. But if I want to send the pipeline to a colleague or attach it as a supplementary material for publishing, then I would go to, I'm losing my mouse. Okay. Hold on. I go to File, Export, Pipeline. And what it does, it basically saves the CP pipe file, and then I can send it to someone else. And when they take the pipeline, like today that we send you guys the pipeline, it didn't have the location of the file or any of the metadata set. So to make it more portable, we save it as CP pipe, and the person who received it needs to adjust and make sure that it reads the images file from the correct location and the metadata, etc. And I already explained. Okay, now that's the fun part. You get to put all of these modules back to back. And when that's done, then make sure to close all the eyes. Again, you can have them go to Window, hide on all windows and run, which is all now hidden. If I click, it's gonna make them all at once. And make sure to have the output setting folder set and know where that is. And then hit Analyze Images after you're done and after you're happy with your pipeline. Any questions so far? No? Ready to go back to the breakout room and start working on the rest?
Sure. Yeah.
Yeah, nice. Did you all get to run the analysis?
Yeah, we were able to.
Yep.
Okay, let's learn a little bit about analyst, and then let's start with analyst demo. I'm just trying to find the correct slide where we are. Okay, now I need to share my screen. Okay, so far, we did detect the objects and we figured how to find them in the image and how to turn them into measurements. What we are interested in is not just to generate a spreadsheet, but to answer the question we wanted to answer. So the question these biologists who ran this experiment wanted to answer again was, what dose of the drug actually caused this GFP translocation factor to translocate from cytoplasm to nucleus? Again, remember our lab motto, to measure everything and ask question later, and this is the later part. We get to answer the question, and it's the most fun part. Based on the features that we selected, we probably ended up with a couple of hundreds of features per cell. Sometimes, depend on the measurement module and the features we care about, we may end up with even over 500 features per cell. So these can end up being a tremendous amount of data. When you have thousands of images, you may have like millions of cells, each with hundreds or thousands of measurements. These are all indicating basically the measurements for each cell. So what do we do is basically treating these results or features like a microarray. So we can look at it, look at all the measurements in a holistic view. It shows for specific measurements that are along on the x-axis, if a specific feature goes up and down on the y-axis in certain cells. This allows us to take a more overall look and see whether certain features are up or down, and then predict certain phenotypes in a specific cells. And all of these we do with CellProfiler-Analyst. With CellProfiler, we made measurements and generated numbers, and with CellProfiler-Analyst, we actually want to look at the features and create phenotypes and answer the questions we care about. CellProfiler-Analyst has several features that are very interesting. You can, for example, look at the histogram or box plots, or look through the data you created already. Once you plot your data, you can take a look and say, oh, this is, for example, an interesting outlier. What do those objects look like? You can look at all the wells in a whole plate and see on average what are the images from those wells look like. You can see, for example, why certain wells look like outliers. Maybe there are scratches in the wells. And the reason our group made this amazing software is because while it's sometimes easy to determine a phenotype from one or two measurements, for example, in the cell cycle, you can typically find from the measurements of DNA content and mitosis marker to see basically where cell cycle phase are your cells in. But there are a ton of biological phenotype that you can't figure out from only one or two measurements that what's going on. All of these are real phenotype from real data. But for example, it's very hard to say the feature that could distinguish these crooked projection from the ones that are longer or more rounder and smooth. The wonderful thing about creating this long list of measurements that you may not be sure like which one to use or which one is important, you can let the computer tell you what is the name of the measurement that is important, and let it tell you which measurements are actually good for distinguishing different classes of what you care about versus what you don't. All of this is called machine learning, which we know is a buzzword right now. But at its core, it's very simple. It's basically putting things into different classes and letting the machine figure the features that distinguishes those classes from each other. Here we have a series of images, and these lines basically represent the different measurements. When an expert, in this case you, looks at these images and decide if that's part of the class that we care about, in this case it could be like these budding niche looking cell projections, they can go through and simply separate the cells into the classes that are belong and versus the ones that are not part of that class. And the computer then will go through and look at the measurement associated with each of these cells in these classes and try to find high values and low values. And then it'll learn from those measurements and it creates a rule. Then you can look at the rule and then you can decide if it's doing a good job or not. And you can do these several times until you can create a good rules that can separate all the objects, or in this case, cells in all of your images properly. And you can leverage these, and the more measurement you create here to make rules out of, you get a better chance of getting a good classification where the computer has learned to distinguish between cells of one group and cells of another. Now let's take the data that you generated and answer the question that we care about, which is finding what dose of this drug causes translocation. Okay, let's now move to demo mode. So now done with CellProfiler. The analyst, I'm just gonna double click, and what it does is basically asking me for properties file. And when you run your analysis, you have this properties file. You just want to locate it and click open. This is a bug for some users that it doesn't show the window, but if you click on the CellProfiler-Analyst again, it's showing you this window. When you get, even though you see this HTTP error, it's right, you're good. But before we move on, I want to show you what actually happened when we analyzed the data. And I want to open this properties file, which is basically a text file. I can see the location of it. If you try to open and it didn't work, you can always check if the location of your file is correct from here. And it has a bunch of information about the image ID and all of the names associated with it. And we can make changes if you need to ever for these. One thing that I'm just gonna try just to show you how easy it is to make changes is to change the color of these. So when I open the images here, it's gonna show me red and green, but I can, since the valid colors are red, green, and all of these, I'm gonna change it to magenta and green. And then we can just hit save and close. We usually would send you to breakout rooms, and so you can again follow along from the PDF file, but because of interest of time and making sure that everybody follows along, I'm just gonna demo. Feel free to ask question if you have question in the middle, and then if you get time, then you can and feel free to also basically follow along, open it, and basically follow the steps with me. Okay, now we have the properties file open. All of these are very useful on the top, all of these in the toolbar, and they are basically available in almost all statistical software, but I'm not gonna go through all of them, but there are information in the documentation of the CellProfiler-Analyst that you can go through. We're gonna test a couple of them. Now let's take a look at the plate viewer to begin with. So what I'm gonna do, this is per image, and this is all the measurements that we basically made in our analysis today. And I'm gonna pick, for example, the intensity of GFP, the mean intensity of GFP in the nucleus. And I could look at the dose, for example, image metadata dose, and we can see that this is how the plate was basically used. These are all my negative controls, positive controls, opposite on column 12, and along row E, we have different doses. This was all in the CSV file that we checked earlier. The nice thing here, you can change how the display looks like. You can change it to thumbnail, for example, and we can double click. Because I changed the properties file after I loaded, it didn't apply the magenta color, but now it's saved with magenta. If I reload it, it would show magenta and green instead of red. We can look at all the images in the wells. That's my positive control that are translocated into the nucleus. A couple of things we can do here is to zoom in, to adjust the intensity, like how brightness it shows, and all of these. I'm gonna go back to the square mode. The most important part here that we are trying to do now is to classify our images. So I'm gonna bring the classifier, and it's not gonna, I can't bring it down. Nope, okay. On the top, you can see that it shows, sorry, I'm just gonna stop a screen share for a second to bring these windows down. If it brings it down, which it doesn't. Okay, let's go back. CellProfiler-Analyst these days like to not cooperate well with me. All right, on the top, it actually asks you, this is what I want you. So it from some random images, I can just bring some random images and look at them and classify them. We want to train this classifier now. We want to do machine learning. So we can fetch, it brings some random images. There is a number showing here, it shows 20. Let me make this bigger. So still not coming down. Okay. Still stuck up there. I wanna bring the image controls and zoom in a little bit so you can see better, and increase the brightness a little bit. To start classification, it does start only from 2 bins. That's the minimum number. But we could add new classes if we want to, and we could change the name of the classes if we want to. We could just right click and say, rename the class. This would be my nuclear translocation. And that would be my cytoplasmic. I'm not going to name the whole thing, cytoplasmic. So then I can look, and here is where basically you can look at only and see, okay, this is nucleus, this is cytoplasm, where is the translocation happening? Another nice thing is when there are several cells or objects in one image, this small square shows which one is focusing on right now. So I'm just gonna drag and drop a couple in each bin. Let's start. We can start from a couple. I'm gonna choose. There are several different algorithms that we can choose here. We know that fast gentle boosting works well here. If you want to learn more about each of those, you can check the CellProfiler-Analyst documentation. And then I can train the classifier. It starts showing me some rules that it basically helps classifying these objects or these 2 groups from each other. Then I can evaluate, and it's gonna give me a confusion matrix that it tells me like the ones that are nuclear and I labeled, or now the classifiers can label it as nuclear, it's actually cytoplasmic. And so 36% is detecting wrong. So we want to make these numbers on the right side, on the proper or correct groups. What is cytoplasmic and really it's cytoplasmic, and what is nuclear are correct, like higher than this. So what I want to do is now to bring more samples and try to train it. And I do this until we are basically happy with the confusion matrix. But that's not the only basically way to say that our classifier works well. We need to test it on images. We need to fetch or bring images from different groups and look at them and make sure they are working well. So what I want to do is just what I said. I'm gonna select all, remove these. I'm gonna just fetch some more and train my classifier more. You see that there are cells, and actually that's how the experiment shows as well. A lot of images, they don't show like any GFP anywhere. I could potentially add a new class and try to have those no GFP in a different class. But I'm not gonna do that right now, but that's another way that you could try. And we could always see what rules are picking. Now it shows mainly cytoplasmic intensity, which is good. If you see the rules, such as the location of like XY location of that cell, that's not the best rule to be picked. Now I want to train again and evaluate one more time. So I can now compare the first one. Now it's doing a much better job. Still a little bit high in this area. And that is where it is actually nuclear translocation, but now we detect it as cytoplasmic. So let's take a look in a different view. I know that these are my negative controls, and I know these are my positive, and these are all in between. So if I double click in one of these, what we can do here is, one thing to do is to drag and drop from one of these to each of the group, which is very nice. Another part is I can say, classify this image. Then I can go to view and say, view classes, cell classes as numbers. So I have two classes. One is show cytoplasmic and the other nuclear translocation. So if I uncheck one, then I can take a look and see if I'm happy with the ones that it picked. Let's take a look. How about the other one? So you see these ones, they really need another group, and they are not really cytoplasmic, but I'm just gonna live with that at this point. I don't wanna just add another class. Maybe I should, but let's just, for the interest of time, let's say that's fine. I'm gonna bring one, do the same, classify image, go to view. It shows me which class is what. So I'm gonna see if any of these cytoplasmic that it chose for nuclear, and I see yes. So I can drag those to my cytoplasmic bin. And then I can train again and evaluate. A little bit better. I'd like to bring these around over 70. So let's now try another way that we can basically look at the data. It's hard to see here, but instead of random, I could bring from, for example, the cytoplasmic group. And before that, I'm gonna select all, remove all, and then fetch from only that group. I can so then look and say, are these any of these that I would call nuclear? Partially, a little bit of signal, but the rest, I'm gonna say it's fine. Select all, remove selected. I could train again and evaluate. I say that's pretty okay, 83, 92%. I could bring another group from the nuclear and check the same, but let's say that's fine. Now what I do is to score all. So it's gonna score all individual objects in all of the images. And it'll create a table. Now I want to save this table to the database table that we had. I can call it anything I want. Hit table, for example. Okay, it asks me if I wanna save it only this one, I wanna store it permanently, and click OK. Now what we can do is to bring another I want to bring another plate viewer, but I'm having a hard time accessing this window. That's what I want. And I can't minimize this. Plate viewer, I'm gonna bring another plate viewer. Now that I have that table part of my database, I can actually just pick that table from the database, and I want a per image. And does this per image is a new set? I say no. So it means that image number in my hit table is similar with the image number in the database that was there. And then from the heat table, I now want p-enriched nuclear translocation. Now let's compare these with the dose. So here it was my negative, and I don't have much of nuclear translocation. That's a good sign. And this is all positive, and I have a lot of nuclear translocation. And I see that it's sort of like a trend here that is increasing at different doses. So this is nice. But instead of having it in two different tables, I want to have and be able to see it in one place. So I click and bring a scatter plot. Then I do exactly the same here. I have per image and image metadata dose. And I know that the dose for this experiment was scaled at logs. So I'm gonna choose log, and on heat table, I'm gonna pick p-enriched nuclear translocation. If I update the chart, I see a very nice IC50. I see a very nice, the log. And then I can say, what is the minimum dose that would cause this translocation to happen? We see that's the highest dose. So still in these doses, the translocation does happen. So then we could say the IC50 is roughly around here, which is a lot less dose than 150 micromolar that was picked or was there before. All good? Any questions here? You see, we answered the question that we started with, or we wanted to answer. See how fun it is to work with analysts. I love analysts.
That's very, very cool.
It is, it is.
In the chat about adding another group for classification. And I sent, basically in the classification window on the bottom right, there's an add new class option where you can go ahead and add as many classes as you want. And I sent a screenshot of that into our Slack channel. So you'll have access to that to take a look at that later.
Can I say one last thing? Sorry, I forgot to mention it's always a good behavior to save your training set as you go. As you do one round of training, make sure to save your training set. Especially that's helpful if you want to end the day and you're not still happy with your classifier, you want to come back tomorrow, you can just easily load your training set, and you can also save your classifier. Sorry, Beth, go ahead.
Yeah. And so for both CellProfiler and CellProfiler-Analyst, you should have access to these videos afterwards from the I2K tutorials website and conference. But we also already have video tutorials available for CellProfiler and CellProfiler-Analyst that you'll be able to take a look at in the future on our YouTube channel. So we'll show you how to get access to those. Okay. I am going to share my screen now, and we're transitioning from 2 dimensions into 3 dimensions for our analysis. And the goal here is that we often get asked this question. People have 3-dimensional data, and they want to be able to analyze their particular biological question in 3 dimensions. A lot of times when you take a Z-stack worth of data, your particular question, the data, if you flatten this into a maximum intensity projection, say, the data then can't actually reveal some really interesting aspect of biology. You need to be able to analyze your full Z-stack of data and consider your data as a 3-dimensional object. And CellProfiler is capable of doing 3-dimensional analyses. The good news for you, since you just went through how to build a two-dimensional pipeline, is that the three-dimensional pipeline is very similar to the two-dimensional pipeline. So what do I mean by that? In a three-dimensional workflow, we're going to start by inputting our data. We'll process the images as necessary using things like filtering to prepare the images for object detection. Once we detect objects, we'll then measure the original images, and we'll save all of our output into spreadsheets, into databases, whatever we need for the further analysis of the data. Again, this is quite similar to the two-dimensional workflow. There are a few differences though, and I want to go ahead and show you some of those differences right now. In the interest of time, I'm going to skip over the slides that I have prepared related to how you input images and extract the metadata. These are all essentially the same as what you do in the two dimensions. You'll have to do a couple of extra steps in three dimensions, but the PDF that we have prepared for you walks you through this. So I'm going to let you go ahead and try it, and then we'll be coming around to the room. So if you run into any questions, Nasim and I will answer them. What I want to jump to is this theoretical consideration. How do we actually approach two-dimensional object detection? And what we want to think about is, what steps does identified primary objects use for object detection? And here I've already answered this question because you just did this. You were adapting the size thresholding for how big can a nucleus be, for example. So you were changing the pixel values for that. You were smoothing potentially the image ahead of time with a filter to make it easier to avoid splitting a nucleus into two when it should be considered as one. You were doing things like applying thresholding mechanisms that were identifying any pixels that were going to be included as part of your image versus the background pixels. And so those were the methods that you were selecting, things like OTSU or minimum cross entropy. Then you were separating apart from each other any touching objects, and you were potentially filling holes in the objects so that you didn't have over segmentation. In two dimensions, you do all of this within the Identify Primary Objects module. The big difference in three dimensions is that instead of using one module, Identify Primary Objects, where you can do these many different things within one module, with three dimensions, what we need to do in CellProfiler is actually split apart these different methods into separate modules. What that's going to look like is that if you need to smooth the image in order to avoid splitting one object into multiple inappropriately, you can do that using a module like median filter or Gaussian filter. Then you would add a separate module if you need to do something like size filtering, where you would measure the object size and shape, and you would filter out the objects afterwards using the filter objects module. For thresholding, you can use the threshold module. For filling holes, we have two different options. In the tutorial today, you'll use the remove holes module. And finally, you go from a thresholded image into individual objects using the watershed module. Other modules in your pipeline are essentially going to be unchanged in their usage compared to how you do this in a two-dimensional workflow. So what I want to do now is just show you a couple of things that you need to know for getting started on your three-dimensional pipeline. And then we're going to send you back into the breakout rooms where you'll get to actually do this yourself, and you can follow the PDF to be able to build a 3D module, a 3D pipeline. Okay. So I'm going to start up CellProfiler here. And are there any questions while CellProfiler is opening up? Sounds like we're pretty good so far. Okay, so for our 3D tutorial, if you go ahead and you have unzipped what we sent you from the Google Drive link, you'll have an images folder that you can go ahead and bring into CellProfiler by dragging and dropping. You'll note that if you click on these right now, it looks like these are the same, it's the same interaction as what you had in 2 dimensions. It doesn't look like a 3D image. So that's not really what we want. The reason is that we have not yet actually told CellProfiler that these are 3D images. We need to do that before we can view these as 3D images. There is an option to extract metadata. In order to get the right regular expression to extract the metadata from these file and folder names, you can open up the PDF. And this regular expression is included here. So I'm going to copy and paste this into my regular expressions. It's split onto 2 lines, so you need to copy both parts. And then you can hit this little magnifying glass, and you can see that the regular expression has taken this file name, and it's able to identify the pattern in the file name to then recognize something that's going to be called the plate, which is here, 3D monolayer. In green, it's recognizing the site. And the most important one for our case is that in blue, it's recognizing what has been told is the channel number. So that's working, which is great. And then we have to hit update. And now we see that each image has been assigned a certain channel number, 0, 1, and 2. Under names and types, what we can then do, this is going to be the most important thing. We have to actually tell CellProfiler, these are 3D images. Once we do that, we can then tell CellProfiler what is the pixel spacing of these 3-dimensional images. And that's going to be based on how the images were acquired from the microscope. You know, what magnification was used, what is the actual distance, the relative distance between pixels in X, Y, and in Z. So for this data, in the PDF, you'll see that the relative pixel is given to you, that it's 0.065 in X and Y, and 0.29 in Z. Finally, at the moment, we only have one image type that's being recognized in our set. And we actually have in our images, we have 3 different channels that all belong to the same image set. So what we see here is we have channel 0, which is a TIFF file, 1 and 2. Since we have now labeled this as a 3D image in CellProfiler, what we can do is when we double click on the 3D image, that will bring up the 3D viewer. And so you can see now we have a Z-slice navigator where we can go up our stack and we can get a sense of what the data look like. We can also use this scroll here to just drag and go down to the bottom of the stack. This channel, channel 0, is the plasma membrane. If we take a look at channel 1, this is a mitochondrial stain. And if we take a look at channel 2, this is our nuclear stain. So you can see most of our nuclei are in interphase, and then we have one mitotic nucleus. Okay, so we have these 3 channels that make up one image set. You'll note that this is actually required for doing 3-dimensional analysis in CellProfiler. Each channel of your Z-stack data must be saved as a TIFF file, and one file that is brought in, a single TIFF file must have only one channel and then multiple Z slices. If, for example, your images are originally an ND2 file type, that you can create a separate pipeline to import that image into CellProfiler and then convert it from the ND2 file to individual TIFF files for each channel. And that's something that if you would need any help doing that, you would want to ask us on the forum, the image.sc forum. We can provide extra guidance. So what I'm going to do here, I'm actually going to let you guys go ahead and finish getting your pipelines set up for looking at the channels. This is something with what we showed you earlier in the 2D pipeline, plus what is included in the PDF here, I think you'll be able to do. And just to give you a sense of what's coming next, then you want to start following this PDF to build a three-dimensional pipeline. And ultimately, your goal is to first, you're going to find the nuclei and create individual nuclei objects. You're going to use those individual nuclei objects to then find the cells and create cell objects. In doing that, you're going to learn some tricks for working with data that look like this, where instead of having a stain that makes the entire cell bright, what's a little tricky here is that we have a stain here where the edges of the cells are bright and the center is dark. So when you find your cells, you're going to learn some tricks for dealing with this type of data. And then once you have the cells, the pipeline will also walk you through making some measurements, exporting them to a spreadsheet, and saving out your data. So that is what I want to go ahead and share with you for getting ready for the 3D pipeline workflow. I think we're probably ready. And if there's any questions, this would be a great time. If anybody has questions about what I showed, I'm happy to clarify anything. But if not, we can probably rejoin the breakout groups and start building 3D pipelines. And how did that go?
Fine.
Good.
We got up to the mono layer.
Perfect.
Section.
Yes.
Excellent.
We also ended up
Good.
in monolayer.
Okay. So since there wasn't time to go ahead and finish the module, I just want to share my screen here, and I can demo and show you the full pipeline and the instructions. Hopefully you'll have some time to finish it out and keep working on the pipeline. We hope that the rest of the instructions should be pretty straightforward to do on your own. So these steps at the beginning, you've made it through. So I just kind of want to walk through. We start by rescaling the intensity for the DNA in particular. This rescale takes all the DNA intensity values and just realigns them from 0 to 1. And that's very helpful in this case for selecting a really good threshold. This is something that we found empirically. We tried to find a good thresholding module without this rescale, and we weren't able to do it very quickly. So we went ahead and rescaled the data. Whenever you use a rescale, be sure that in your later measurement modules, you focus on only measuring images that are the original images rather than your rescaled data, just to keep everything accurate. We then did a resizing. So this is a step you all made it through, and that helps to downsample our data, which does two things. It'll make processing times much quicker, because instead of dealing with a 0 to about 250 pixel by 250 pixel by all of these planes image, you've got one that's a lot smaller. It's half the size. The other thing that happens is that when you go down and you make something half the size, it can kind of smooth out some of the pixel intensity values, because it kind of averages everything with its neighbor, which is creating essentially one smoothing step on the image already. We then take that a step further with this median filter module that you guys saw. So you can see in our filtered image on the right, we've really smoothed a lot of the intensities in the nuclei, which is going to help prevent any oversplitting of the nuclei when we do the later steps. We apply a threshold. You can see there are some holes where within a nucleus, there's these pixels that weren't identified as being above the threshold that was automatically selected. If we leave those holes, then that can lead to errors later where a nucleus will be inappropriately split in half because of the hole in the middle. So that's why we use this next module called the remove holes module. And now you can see how these little dark spots have been filled in. Finally, we do the watershed, and this is where we're actually going ahead and going from our thresholded image into separated out individual nuclei. And if we go through the Z planes here, you can see that these nuclei, all the planes kind of assign the nuclei to the appropriate object. So here in slice 37, these pixels are identified as part of one object that is the same object as when I go down to slice 36. So that's the process of selecting the nuclei. We can then resize them back up and we get to the full size image, just like what we started, and we've identified our nuclei. Are there any questions at this point before I go forward? Okay. We only have a few more minutes, so I'll try to walk through this quickly. The basic idea for what we're doing next is that we need to go from those nuclei, and we're going to do the same thing that we did in ID secondary objects, where we go from a seed to then expand outward and find the boundaries of the cells. As I mentioned, this data is a bit tricky though, because instead of having an image where the center of the cell is stained brightly, we have images where the center of the cell is dark and the outside edges are bright. And so what can you do if you have that? The first step to finding the cells is that we have to create seeds for them using the nuclear objects. And we're going to start by shrinking the nuclei to make them a little bit more seed-like. So we go ahead and do that on the small nuclei. We shrink them a bit more, and now they look a little bit more like seeds. From there, we're going to resize those back up to the original size. And we're going to convert them from an object into an image. And this is the format that we need when we're going to do a watershed module later to identify the cells. Now we're going to start manipulating our membrane image to try to get to something where the center of the cell is bright and the edges are dim. So we start by going ahead and thresholding the membrane image. I think many of you saw this threshold. From there, we invert the image so that now this looks pretty good. We've got a center that's bright, the edges are dark. But if we scroll down to the bottom, what was background in the original image is now very bright in our thresholded image. And that's not what we want. We want the background to still be black. So how can we do that? One thing we're going to do here is we're just going to first fill the holes in this image. That's unrelated to the background problem. That's just getting this image closer to what we want for having bright centers. And now we're going to start addressing that problem of trying to get rid of those bright backgrounds, trying to get rid of this bright background part. So we start by adding together all of our images. This will give us something where we can identify where there are cells versus where there are not cells. We do a resizing module to make this smaller, so we shrink it into by 4. That's going to help again with computational time, and it's going to make the it's going to smooth the image in a way that's very helpful. We use this closing module that gives us something that we can use to actually threshold and identify where there are cells versus where there are not cells. We go back up to the original size and we apply a threshold. And this threshold now, it's bright where there are cells. And then if we go all the way up to the top of the Z-stack, you can see it's dark where there are not cells. So bright where there are cells, dark where there are not cells. We use the mask image module to actually apply that to our original mask thresholded image. And now you can see we've fixed that problem. So we have bright cells, and then we go to the top of the image and it's black as we expect. Here, we're going to just erode the image to help preserve the boundaries between cells a little bit better. And then we apply this watershed module, which gets us to our final cells image. Here on, you can see the segmented cells, and you can see we've got nicely segmented cells to go along with our segmented nuclei. And the rest of the pipeline is things that you've seen before. We're going to measure the objects. We can create some images so that we can output them and see what it looked like. But that is essentially the 3D pipeline that we wanted to share with you.
Closing thoughts and final notes. We just want to introduce the Image Analysis Forum, which plays an important role in assisting the biologists in using software for image analysis. It's currently each open source software. They used to have their own website and email list, but recently the groups leading CellProfiler and ImageJ project teamed up to merge their forums toward creating a collaborative network of support for the scientific image community. And the majority of bioimage analysis softwares are already joined and they are part of the forum. So if you have any questions, feel free to post on the forum, search the forum, make sure that you tag us, like tag CellProfiler in here or any other softwares that you have questions from. It's a great source. And the collaboration of CellProfiler and ImageJ project this year led to a center grant for open bioimage analysis, or COBA. The project started this year and the website is up and running, and we started on several projects already. Check the resources that are available to you, the forum, COBA YouTube channel, and examples on the website to get a sense of the support network that is already exists. You will find that image analysis can be complicated, can be tricky, and sometimes it takes a lot of people to solve a problem. But the community is here to help you, basically. And last but not least, we want to thank our lab members. Pearl was not here when we were taking this picture, so we added her picture here. All of the Ann Carpenter lab members in imaging platform abroad, supporting CellProfiler and CellProfiler-Analyst, and a couple of exciting softwares coming up as well. And many thanks to our biology collaborators and the grants that we have. Thank you all. Thanks for listening. Thanks for staying with us. It was a wonderful experience.